Now we get a closer look at the most important tissue of the eye. That's the retina. So that's the innermost membrane at the back of the eye. It's called the retina. The retina is more than one layer of cells. The most important light-sensitive cells, there are two kinds. One is called the rod, the other is called cone. So it's cone cells and the rod cells. The shape is quite peculiar. They're mostly bipolar. The cell body and one end, they have a terminal synapse onto the bipolar cell. On the other end, they form just like a comb, like the comb you brush your hair. That is, they increase the cell membrane region so that they can catch up more light that gets into the eye. So this is all for the purpose to catch light for us to see. The cone and rod cell are specialized receptor cells. They are not the real neuron. From there, they will synapse with the interneuron bipolar cells. So these bipolar cells, one synapse in contact with the cone and rod. On the other hand, they will synapse with the ganglion cells. These ganglion cells, their axon will form the optic nerve that goes into our central nervous system, into the brain. And also, in between the cone and rod cell and the ganglion cell and bipolar cell, there are two other types of cells. One is called the horizontal cells, and the other is called the amacrine cells. It's like glial cells in the brain. They help to maintain the surrounding environment within the eye. And there's another thing probably I should mention that if you say it's a design, the actual forming of this retina is not very ideal, because you see the light-sensitive cells is actually at the very back of the eye. The light has to pass the ganglion cell, pass the amacrine, pass bipolar cell, pass horizontal cell, then reach the real light-sensitive rod or cone cells. It's not very efficient, first of all. And then the second one is the retina touch with the base of the eye. There's a pigment epithelium, and then there's a choroid, and then there's a scleral membrane. And there's one serious problems when you get older or you get injured of your eye, or some people with a very high degree of nearsightedness, so their eyeball may get a little bit deformed. In that case, the retina will suffer detachment, because they are not contact with these pigment cells. Really is not that secure, not a very good design to us. The stimulus of light go all the way, hit the retina, that's rod and cone cells. Then the information will transmit to bipolar cells and to ganglion cells. And then all the axon of the ganglion cell form the optic nerve. These optic nerves, they will go through the base of our brain, and some of the optic nerves will cross to the other side of the brain, so it's called optic chiasma. Some will stay. Once they get into the brain, we call the optic tract, and they will make one relay, one stop in the thalamus first. And there's a special nucleus called the lateral geniculate nucleus on each side of the thalamus. And from there, they will project all the way to our back, the visual cortex. And from the visual cortex, that's the first station. And also they will send information to other visual association cortex. So this is the neuroanatomical pathways. These photoreceptor cells, cone cells or rod cell, is a very peculiar type of cells. The blue arrow shows the process favored in the dark. The orange arrows just shows you when there's a light. Why I say it's a peculiar type of excitable cells? It's not a real neuron, it's a receptor cell. So it can be excited just like neuron. Usually the receptor cells without stimulation, they are quiescent. When stimulated, they become activated, but not photoreceptor cells. During darkness, photoreceptor cells is activated. That means it has action potential, always going on. That's all because during the darkness, the granulocyclase will convert GDP into cyclic GMP. So cyclic GMP will stimulate the cation channel, so let the sodium calcium flux in. So if you remember, when the sodium gets in, the membrane gets depolarized. So there will be action potential. When there is light coming in, they will hit special photopigment. So that the photopigment determines this photoreceptor cell sensitive to which wavelengths. There's a molecule called the retinol. When the light hits the photopigment, it will go through G-protein process, goes through several subunits. The one is called the opsin and the other is called transducin. It will activate cyclic GMP phosphodiesterase, convert cyclic GMP to just GMP. So in this case, there's no more cyclic GMP, so the cation channel will be closed. So the membrane will stay quiet, will not be depolarized, will be repolarized. And then actually in this case, the photoreceptor cell send out the signal. So that's totally different from most other receptor cells. During rest, it's actually activated. And during light stimulation, it's just been deactivated. And let's get a little bit discuss of how do we see light. That's all because this rod cell or cone cell, they are sensitive to the wavelengths of the visible light. And the rod cells only respond to 500 nanometer wavelength light. They cannot differentiate between colors. So we just say they are black and white cells. They are very sensitive to light. If there's total darkness, someone strike a match, light a cigarette, someone can see it from miles away. So that's how sensitive our rod cell is during the night. And then the cone cell, there are three kinds of cone cells. They all have their sensitive region of the wavelength. So one is the red cone cells. They are sensitive to longer wavelengths, so they sense the color of red. And then we have green cone cells. They are sensitive to the middle region of the wavelengths. And then we have blue cone cells. They are sensitive to shorter wavelengths. So in combination of different cone cells being stimulated, we see all kinds of colors. We can see many different kinds of colors. Just give you an example. This is more or less like a blue cone cell. Because the ganglion cell through bipolar cells and the bipolar cell through a cone cell. So the ganglion cell itself is also determined by the type of cone cell. In this case, this is the blue sensitive cone cell. So when the blue light is on, you can see the action potential in the ganglion cell increased. When it's red light, it's actually decreased. Even white light, not as much response.